we have one more major category of inheritance to cover, and then we're going to look at some meiotic errors. We need to talk about what happens if the inheritance happens on the sex chromosomes, not the autosomes. So everything we've talked about so far has involved the autosomes, chromosomes 1 through 22. What about the sex chromosomes? Well, that type of inheritance is a little bit different in that gender matters. The other types of inheritance that we've talked about, autosomal inheritance, it doesn't really matter which parent gave you the disease or the genetic defect. And the gender of the offspring isn't relevant. But now gender is going to be relevant. We're going to talk about X-linked inheritance. It's sometimes called sex-linked, but we're talking about inheritance on the X chromosome. Remember, everybody, if they have a normal array of chromosomes, everybody has an X. So if you're XX, you're female. If you're XY, you're male. And then we're going to see when we talk about trisomies and monosomies, you're going to see that there are some differences that can occur. But for the most part, everybody has an X chromosome. You have to have an X in order to be alive. You don't have to have a Y in order to be alive, but you have to have an X. That's because the X codes for a lot of things that have nothing to do with being male or female. I've got a slide that's back at the very beginning, so sorry for this, but I'm going to have to skip all the way back forward again. Here we go. This is a partial map of the X chromosome and the disorders that are carried on this chromosome. And most of these have nothing to do with being male or female. In fact, all of them have nothing to do with being male or female. Okay, so this list here, hemophilia type A, G6PD deficiency, drug-sensitive anemia, chronic hemolytic anemia, colorblindness. The list goes on and on. All of these disorders are carried on the X chromosome. Both males and females have an X. But here's the big difference. If females have a defective gene on one of their X's, it's not necessarily a bad thing for that female. If she has a normal copy of the recipe, she can still make that protein correctly and can probably make enough correct protein to overcome the fact that she has a defective protein. If this guy has a bad recipe on his X, does he have another normal copy? No, he does not. He does not have another X. So in this case, he would have the disorder. He does not have another X chromosome to make that protein correctly. Therefore, these X-linked disorders tend to occur more in males because females can actually be carriers. Males cannot. There are no male carriers in this story. If he has that gene, he's got the disorder. So in this case, gender is going to matter. Two classic examples, colorblindness and hemophilia. And in order to indicate a normal X versus an X with a defective gene, we'll use colorblindness as our example. And here are our possible alleles. So two normal X's, we're going to call this a normal female. She does not have the disease allele on either of her chromosomes. X with a superscript C. This female carries the colorblind allele on this X chromosome. She would be called a carrier female. The only way this female would have colorblindness is if both of her X chromosomes carried the colorblind gene. This would be a colorblind female. Males only have two possible genotypes. normal, meaning no disease, normal copy of that gene. If his X has the colorblind allele, he will be colorblind. He does not have another normal copy of that recipe. So this would be a colorblind male. Let's do a couple of crosses so I can show you what I mean by now gender matters. Gender of each parent matters. Gender of the offspring matters. Let's do normal female with regards to colorblindness times colorblind male. Okay, what would the genotypes be? Normal female means no colorblind allele on either X. Colorblind male, that's his genotype. Again, the gender of the parents and the gender of the offspring only matters when we're talking about chromosome pair 23. Not autosomes, but the sex chromosomes. So let's do our cross. Punnett square. Doesn't matter which parent you put where. We'll do mom here, dad here. Okay, let's do our crosses. This cross is so interesting to me. I mean, obviously, it is what it is. It's math. But it's not possible to have a son with color blindness in this cross, even though dad was colorblind. That's pretty crazy. So we end up, all of our female offspring are carriers. So now, remember, gender of offspring and gender of parents matter. These are all our females, right? XX babies are females and XY babies are males. So these are our female offspring and these are our male offspring. So females, 100% carriers. Males, 100% normal. So even though dad had color blindness, none of the male offspring have it, but the females are carriers. What if a carrier female mated with a colorblind male? So, carrier female times colorblind male. So our carrier female, what is her genotype going to be? She's going to have one X that's normal and one with a colorblind allele. Remember, no such thing as a carrier male. He either has the disease or he doesn't. So there's our colorblind male. Mom, dad. Oops. This is interesting because we really end up with one of each. We have a colorblind female and a carrier female. So just looking at our females, 50% colorblind and 50% carrier. Again, these are our female offspring, XX. These, of course, are our male offspring, XY. And of our males, 50% are colorblind and 50% are normal. That's interesting too, I think, because carrier mom and colorblind male, You wouldn't think it would be possible to have males that didn't have colorblindness. But 50% of our male offspring don't have it, 50% do. So that's how X-linked inheritance works. And again, it's because males don't have another copy of that X chromosome. If they get the defective gene, they've got the defective gene. There is no normal recipe for how to make that protein. And again, that's because the X chromosome codes for a lot of things that have nothing to do with being male or female. So this is all related to meiosis. I know on the surface meiosis probably didn't sound that exciting, but it actually really is, because this is how you got one allele or the other from your parent. So each time this colorblind male makes sperm, on average 50% of his sperm are going to get the colorblind X, and 50% are going to get this normal Y. And then the same thing goes for the mom. What happens if there's an error in meiosis and we don't end up with the correct number of chromosomes? Well, let's look at two examples of this. This can happen in meiosis 1, or it can happen in meiosis 2. And it's called Nondisjunction. It's when the chromosomes don't separate correctly during meiosis. Nondisjunction. It would help if I spelled it correctly. Failure of chromosomes to correctly separate in meiosis. It can happen in meiosis 1 or meiosis 2. And it can happen with eggs or with sperm. We do know that there's a correlation between female age and the incidence of nondisjunction. Why? Well, that's because when females are born, all of their egg cells are already present in the ovaries and have reached prophase 1 of meiosis. Then every month, one of those egg cells will make it to maturity and will continue to go through meiosis as it's maturing. But females are born with all of their eggs already at some stage of meiosis. Males are constantly making sperm. Males can have nondisjunction occur also, but there's not a correlation with age. The chances of doing it at age 16 are the same as at 16. So let's look at this example. Remember meiosis 1, homologous chromosomes separate, each still having 2 chromatids. So this is chromosome 1, chromosome 2. These don't pull into 2 separate cells, so this cell gets way too much DNA and this gets not enough. So by the time those cells go through their second division, these 2 cells ended up with an extra one, and these ended up 1 short. If you end up with 1 of a chromosome, remember you're supposed to have 2 of each, it's called a monosomy. 3 of a chromosome is called a trisomy. Monosomy and trisomy. Let's look at, this had happened normally in meiosis 1, and then in meiosis 2, these didn't pull apart. Same thing. You're going to end up with 1 cell that has too much DNA and 1 that doesn't have enough. This would be the normal condition in. Okay, so 1 of a chromosome is going to be N minus 1, and extra is going to be N plus 1. N is the normal. Nondisjunction. Here's the rule. There are no living autosomal monosomies. You have to have at least 2 of your autosomes. Remember, autosomes are 1 through 22. There are no living autosomal monosomies. There are some living autosomal trisomies. These happen to be the chromosomes that code for the smallest number of traits. If you had 3 number 1s, you would never develop. So those trisomies do occur. Those babies are just not born. There's usually a spontaneous miscarriage during the first trimester. Sometimes you might not even know you were pregnant with that baby. But those babies never develop. It's too many proteins that are not being developed correctly, that are not coded for correctly when you have 3 copies of that gene. 21, 18, and 13 happen to be chromosomes that don't code for as many traits. 21, if you have 3 21s, you have Down syndrome. This is incorrect. It should just be Down syndrome, not Down syndrome. But trisomy 18, Edwards, and trisomy 13, Patau syndrome. So 13 and 18 have some severe birth defects. 21, whole range of abilities of these people. Some have fairly normal lives. Some have more issues than others. So there are some survivable autosomal trisomies, but no living autosomal monosomies. The sex chromosomes are a different story. Okay, you can have disorders where you have an abnormal number of sex chromosomes, and otherwise you seem to be normal except for sex traits, possibly. You have to have an X chromosome. You can't just have a Y. Again, that X codes for a lot of things that have nothing to do with being male or female. So Klinefelters, these are boys that have 2 X's. So something went wrong during meiosis, and they ended up with 2 X's from mom. The egg that made them had 2 X's. The Y is obviously from dad. Okay, then we have XYY males. Well, that happened in dad's sperm. Too many Y chromosomes. We have triple X females, meaning that they have 3 X chromosomes, and these are metafemales. They're often taller in stature, but otherwise pretty normal. And then we have Turner syndrome. You can have just 1 X chromosome and didn't get a chromosome from dad or mom, whatever the case might be. The egg or sperm didn't give you a 23rd chromosome. But you can't be just Y. So that's what happens when chromosomes fail to separate correctly during either meiosis 1 or meiosis